Chapter 25, Seedless Plants. Let's look at early plant life, green algae, bryophytes, and seedless vascular plants. Seedless plants used to rule the world with a force. They covered almost every inch of the planet, and their slow decomposition over thousands and thousands of years made the coal deposits that we're burning and worrying about right now. As you know, those plants are carbon-based. So with each carbon-based fire, we release carbon dioxide into the atmosphere, with fewer and fewer plants to take it back out of the atmosphere as forests are clear-cut and grasslands are paved. As with a fluffy coat in winter, the added layers of gases are increasing our global temperatures at an extremely dangerous and continuous pace. Let's look back on early plant life before this carbon sink had a chance to form. Kingdom Plantae is large, with about 300,000 species. 260,000 of them have or regularly form seeds, so we're going to look at that other 40,000. As plants moved on to land, they faced both some problems and some benefits with this new shift. For the problems, drying out was a constant concern. They were used to having water for structural support, to filter out dangerous radiation from the sun, and to help their gametes move, so they had a lot of obstacles to overcome. However, there were a lot of benefits. Sunlight's more abundant above water, carbon dioxide's more abundant, and because they were moving away from their predators, they had a lot of time to evolve without any concern for predation. There are some more specific structural advantages for plants on land. One of those structural changes that occurred was the alternation of generations. Here's the basis of that system. Zygotes of plants, which come from two sexually different gametes, fuse together and grow via mitosis into a 2N sporophyte plant. The sporophyte lives like a normal mature plant and eventually produces plant spores via meiosis. A single individual spore can grow into a 1N gametophyte plant, which looks like a normal plant too, it just so happens to be 1N. It's capable of forming some gametes for sexual reproduction. Some plants spend more of their life cycle in their gametophyte form, and we refer to them as haplonic plants because they're haploid most of the time. Diplonic or diplonic plants remain sporophytes for longer. There are even some plants that spend an even amount of time in both their gametophyte and sporophyte forms, and we call them haplodiplonic plants, which is a nice mouthful. A second major advantage of land plants is the sporangia in seedless plants. Sporangia are found in sporophytes, and the term literally means spore in a vessel. They're reproductive sacs that contain 1N spores that are created by sporocytes. These are just mother cells that are made for undergoing meiosis. Plants are capable of creating two different types of spores. One type of spore are called homospores, and they're generally formed in seedless non-vascular plants. They're made in the gametophyte dominant individuals, and they have male and female plant parts forming on the exact same plant. So they produce only one type of spore. Heterosporous plants tend to be seedless vascular plants. These plants produce two different types of spores. Some of them are male and they make microspores, which are smaller, and some of them are female and make megaspores, which are larger. Both types of spores are protected by a tough polymer known as sporopollenin, which is a mixture of fatty acids and carotenoids. Carotenoids is a pigment that turns things yellow, and that's why most pollen is yellow. The covering is resistant to chemical and biological degradation, hence the numerous pollen fossils that can be found if you know where to look. Let's take a closer look at the male and female plant parts. Gametangia are structures observed on multicellular haploid gametophytes. Precursor cells give rise to gametes via mitosis. The male gametangia is known as the antheridia, and it creates sperm that swim to the archegonia, or the female portion of the plant. The growing embryo develops inside of that archegonium. A final structural improvement for plants on land were their shoots and roots. Because the shoots and roots of plants increase their length via division in a tissue referred to as the apical meristem. The apical meristem contains undifferentiated cells that divide rapidly and become specialized as they grow. Quick movement towards resources allows plants to make the best of their given environment when the base of their root system is anchored to the ground. There are some additional land plant adaptations, like the evolution of vascular tissue. We'll look at the specifics of vascular tissue a little bit later. They also developed a root system for water and support, a cuticle with stomata for gas exchange and to help stem water loss. They have different pigments that can absorb and protect them from UVA and UVB light forms. They have developed chemical defenses, spikes for defenses, or sweet fruits to nectar when they would like to draw a mutualistic partner near rather than push them away. The evolution of land plants occurred during 6 different periods of time. The book runs quickly through them. I would just like you to understand the breadth of the time elapsed during the evolution of plants. We'll see a lot of changes, but it's because we're looking at a massive expanse of time. You can refer to all of the land plants as charophytes or as embryophytes, and then they are either non-vascular or vascular in nature. Non-vascular plants that are seedless are called bryophytes, and there are 3 major groups of them: liverworts, hornworts, and mosses. We'll look more closely at these a little bit later in lecture. The vascular plants can either be seedless or seed-bearing plants. The seedless plants are called lycophytes and pterophytes. The lycophytes are club mosses, quillworts, and spike mosses. The pterophytes are whisk ferns, horsetails, and regular ferns. We're going to focus on seed plants next week. Now that we know where we're going, let's take a step back and look at the precursors to land plants, the green algae. We used to clump all photosynthetic eukaryotes in the same group of kingdom Plantae. Brown, red, and gold algae, as we now know, are all considered part of kingdom Protista. Green algae is a little bit harder to place. While they have some of the same pigments, they store their energy in the same way, and they divide their cells in a similar fashion to plants, they're missing some of their characteristically plant-like features. We now lump land plants and most of green algae into a subphylum called the streptophytes. The remaining green algae belongs to Chlorophyta. There are about 7,000 species in this group, and the members are seen here. They live in fresh, brackish, and salt water and some damp environments, including snowpacks, which shows you how highly evolved they are, alluding to their evolutionary age. Some green algae within the subphylum Streptophytes belongs to the order Pterales. Their evolutionary history can be traced back to about 420 million years ago. They range in length from millimeters to meters. They tend to have a characteristically bad smell and 2 major cell types. Large cells form their thallus, while the nodes along those cells are made from smaller ones, with female and male reproductive structures attached. This order does not undergo the alternation of generations, but it does have other characteristically plant-like features, like lignins for support, spore pollen protecting its spores, and plasmodesmata between the plant cells. Now we're going to focus on the bryophytes. They're the closest living relative to extant early terrestrial plants. Little structural elements to them, there are few fossils that we can study. There are about 25,000 species of bryophytes known today, and they tend to thrive in various damp habitats, although a few have adapted to our deserts. All of their conspicuous vegetative organs belong to the haploid version of the organism, known as the gametophyte. You don't really notice the sporophyte as it's attached to the adult version of the plant, which nourishes it as it grows. There are 3 phyla of bryophyte. The first phyla is the liverworts. Liverworts colonized nearly every terrestrial habitat on early Earth, and nowadays there are about 7,000 existing species. The lobes of the leaf-like structures are shaped like the human liver, which is where their name came from. They do have openings in these leaf-like structures that look like stomata, but they don't open and close like stomata do. They also have female gametangia on different plant forms. Here's an example of those liver-like lobes as seen in this particular species of liverwort. Here you can see the life cycle of the liverwort, which can occur entirely on the same plant. Down below you can see the antheridial head on the male plant, that is 1N, and the archegonial head on the female plant, that is 1N. The sperm created within the antheridial head can swim to the archegonial head and create a 2N embryo. The embryo grows on a slender stalk that we call a seta that exists entirely on the gametophyte plant. It has spores created via meiosis that are growing within the thallus. Rhizoids, or root-like structures, help to anchor this entire plant to whatever surface it's attached itself to. When matured, the spores can blow away or be moved by water to a new location to create a whole new gametophyte plant. Hornworts are the second group of bryophytes. In hornworts, the short blue-green gametophyte phase is dominant. They're easy to pick out because they have these narrow pipe-like structures. They have developed stomata and also contain a singular chloroplast in each of their thallus cells. They do have meristem cells that can help them increase their height even though they're without vascular tissue. Here you can see the alternation of generations. They too have a large gametophyte plant that's 1N. To me, it looks like a piece of lettuce almost. The thallus is this big flat body, and it has rhizoids or root-like structures that help attach it to the ground. It has an archegonium that contains the 1N egg and the antheridium that creates the 1N sperm. The sperm swim into the archegonium and fertilize the egg, producing a 2N embryo. The 2N embryo can grow into a tall, slender sporophyte where meiosis will produce more 1N spores. Each 1N spore can grow into its separate gametophyte plant, which just continues the cycle. Let's move on to the mosses. There are about 10,000 species of mosses, and they have widely varying habitats. They're helpful because they slow down erosion and store moisture and soil nutrients for the plants around them. Mosses can be hard to grow, however, because they're very sensitive to copper salts. They have diminutive gametophyte forms and some really great adaptations for dry land, like the stomata and the rhizoids previously seen in a bryophyte. The moss life cycle follows the pattern of alternation of generations, as seen on this slide. The most familiar structure is the haploid gametophyte, which germinates from a haploid spore and forms the first protonema, usually just a tangle of single-celled filaments that hug the ground. Cells akin to an apical meristem, like we saw in the root-type structures, actively divide and give rise to the gametophore. This consists of a photosynthetic stem and then some foliage-like structures. Rhizoids, root-like structures, form at the base of the gametophore and help to anchor it to the ground. The gametangia of both sexes will develop on separate gametophores. The male organ, that's the antheridia, will produce quite a few sperm, whereas the female archegonium only actually makes a single egg. At fertilization, the sperm swims down the neck to the venter and unites with an egg inside of the archegonium. The zygote, which is nice and protected, divides and grows into a sporophyte, which is still attached by that foot down to the gametophyte. So 2 different generations are capable of growing in one location, essentially on the same plant. Vascular plants, or tracheophytes, are the dominant and most conspicuous group of land plants. There are 260,000 species of seedless vascular plants, and they represent more than 90% of Earth's vegetation. Several evolutionary innovations explain their success and their ability to spread to all habitats. For instance, they have formed vascular tissue, known as xylem and phloem. Xylem is the long-distance transport tubes that move around water and nutrients, while phloem transports larger macromolecules, like sugar, proteins, and other important solutions and solutes. Vascular plants have developed true roots. They help to support the plant and draw in water. And finally, they have leaves, sporophylls, and strobili. Sporophylls are leaves that can bear sporangia, and strobili are cone-like structures that contain sporangia. You might know these as pine cones. We're going to study 4 phyla of seedless vascular plants. The first phyla we're going to look at are the Lycopodiophyta, or the club mosses. These are one of the earliest groups of plants, and they're small and evergreen. They consist of just stems and some microphyls. There's about 1,200 species of them, and they do undergo an alternation of generations. For them, the sporophyte phase is their dominant phase. They're also capable of creating 2 different spores, homospores or heterospores, which allows them to increase their genetic variation. The second phyla includes the horsetails, and they have stems that are characterized by their many joints or nodes. They also contain needle-like leaves, so photosynthesis occurs in or on their stems rather than in or on their leaves. They have silica in their epidermal cells, which helps them to be extra firm and grow quite tall. Rhizomes help to anchor the whole plant to the ground. Here's a close-up picture where you can see the thin leaves that are radiating from individual joints that are quite noticeable on horsetail plants. Horsetails are so firm they were used as scrubbing brushes, like something you'd use to clean a kitchen pan. A third phyla is the phyla that contains the whisk ferns. They lack true roots and leaves, which were probably lost somewhere along their evolutionary path. Photosynthesis occurs on their stems, and the yellow nodes that you see in this picture are just little sporangia. A final group to study are the ferns. There are about 20,000 species of ferns, and they have a really wide range of habitat. Most prefer moist, shaded areas, and they are sporophyte dominant. They have particularly large fronds, where they both undergo photosynthesis and hold their reproductive organs. They have some plant parts that may grow in strange places, so you see multiple stems and sometimes roots sticking out from stems that are kind of far from the ground, but it seems to work for them. Here's some close-ups of fiddleheads, or the tips of fern fronds. They can curl into some pretty interesting shapes, and it means that a lot of people cultivate them just because they're kind of neat-looking plants. Here you can see the life cycle of the fern, which shows an alternation of generations, with a dominant sporophyte stage. Hopefully by now, this picture looks really familiar, so I won't take your time to walk you all the way through it. Most ferns produce the same type of spores and are therefore homosporous. The diploid sporophyte is the most conspicuous stage of its life cycle. On the underside of mature fronds, sori, or a singular sorus, forms as a small cluster where the individual sporangia develop. Inside of the sori, there are spores that are being produced by meiosis and then get released into the air. Those land on the ground, and if it's a suitable substrate, they'll germinate into a heart-shaped gametophyte, which is attached to the ground by a lot of really thin filamentous rhizoids. Here you can see that inconspicuous gametophyte that harbors both sex gametangia. Flagellated sperm release from the antheridia and swim across the wet surface to the archegonia, where the individual eggs are fertilized. The newly formed zygote grows into a sporophyte, then emerges from the gametophyte and grows by mitosis to form the next generation. Again, hopefully by now, this sounds a little familiar. Ferns contribute to the environment by promoting the weathering of rocks, the acceleration of the formation of topsoil, and by slowing down the erosion by spreading their rhizomes into the soil. They not only help anchor themselves, but they literally help anchor our planet. Water ferns of a particular genus actually help to fix nitrogen because they're great space for nitrogen-fixing cyanobacteria to survive. So they help to restore depleted nutrients in aquatic habitats. Seedless plants have historically played a role in human life because we use them as tools, fuel, and medicine. Dried peat moss is a commonly used tool in some parts of Europe and is a renewable resource. If you've ever seen Monty Python and Knights of the Holy Grail, that's what the peasants were digging up in the field. If you've ever seen mosses put on top of potted plants, it's because some florists use blocks of moss to maintain moisture in floral arrangements. But by far the biggest impact of seedless vascular plants comes from their extinct progenitors. The tall club mosses, horsetails, and tree-like ferns that flourished in the swampy forests of the Carboniferous period gave rise to the large deposits of coal, which is where we started class. Coal provided an abundant source of energy during the Industrial Revolution, which had tremendous consequences on human society, including rapid technological progress and the growth of large cities. However, it's also greatly contributed to the degradation of our environment. Coal, unfortunately, is still a primary source of energy and a major contributor to global warming. With technology, innovation, and research, hopefully we can move away from coal as something that's destroying life on our Earth to things like wind and solar power that's already here, 100% renewable, and not at all dangerous to the world around us. With that, we'll end our discussion of seedless plants. Go now to read the chapter, write notes in your own words, do practice problems in OpenStax, and do the practice activities that are there for you in Learner, and then complete your homework assignment, also found in Learner.